Hello everyone, and welcome to the first MDPI webinar on water, which is entitled Sewage Screening as an Early Outbreak Alert Tool and SARS-Covid-2 Fate in the Aquatic Environment. Thank you very much for joining us today. This is the first webinar of a series based on the special issues and sections from our journal Water. If you are interested in submitting your paper to a special issue, a list of all the special issues are available on the webinar webpage. This webinar will be chaired by Professor Adriano Bordalo from the University of Porto, and will have 4 presentations given by Professor Bordalo himself, Dr. Erika Gavis, Dr. Jennifer Guidas, and Professor James van der Slis. This will be followed by a discussion and a Q&A session moderated by Professor Bordalo. And if you have any questions, please feel free to post them in the Q&A box. I would also like to remind everyone that this webinar is being recorded. The recording will be posted on the webinar webpage for everyone to watch it as soon as possible. And we also have YouTube live streaming. So finally, just to present our chair, Professor Adriano Bordalo is an associate professor in the Institute of Biomedical Sciences from University of Porto in Portugal, with 40 years of experience in the field of environmental and clinical chemistry and public health. He has published over 130 peer-reviewed papers and book chapters on different fields of life and health sciences. He has also served as a scientific consultant in West and East Africa and in universities from Eastern Europe, Central and Southeast Asia. I will hand out the microphone to him. Thank you, Adriano, for accepting to chair this webinar, and everyone, enjoy.
Okay, thank you very much. Welcome, everybody. First of all, I would like to thank Water and the staff involved in the organization, dissemination, and implementation of this webinar, dealing with a subject that somehow has kept the attention of the health authorities worldwide. Therefore, this is the right time, and MDPI understood it. I'm quite glad. Thank you very much again, and I would like also to extend my compliments to my co-speakers today, Dr. Erika Gavis, Dr. Jennifer Guidas, and Professor James van der Slis. The present coronavirus pandemic has an unprecedented impact on the daily life worldwide, since the first human cases were reported by the end of December last year. At the onset, the virus was introduced to humans through contact with infected animals, as demonstrated by early molecular studies of genomic sequences. The international response was overwhelming, engaging all sectors of the society. Indeed, the pandemic fostered an unparalleled response from the scientific community to find answers to so many unanswered questions. Actually, we are in the presence of a novel coronavirus with somehow different characteristics from the cousins SARS and MERS that were behind the smaller scale events in 2002, 2003, infecting 8,000 people in 26 countries in the case of SARS, and the still ongoing epidemic, but quite forgotten MERS epidemic that started in 2012, affecting 24 countries and yielding 2,494 cases up to November 2019. These are the latest stats, official stats from the WHO. The majority of MERS cases have occurred in healthcare settings, and 80% of the cases in a single country. Our novel coronavirus showed a faster dissemination rate than its 2 previous ancestors. As a result, the distribution magnitude of infected populations is the most fundamental aspect of this current pandemic. Originally, we are in the presence of a zoonotic virus that was able to enter human populations via direct or indirect contact with infected animals, like bats, dromedary, pangolins, for instance. However, the exact route of transmission from animals to humans is not yet fully understood. Well, once established in the population, person-to-person transmission may occur. In the case of the current SARS-CoV-2, 2 major direct routes were initially identified through air droplets, face-to-face contact with a sneeze or cough, and surface contact with secretions of people who are infected. Since then, an increasing number of studies show that may be shed through multiple routes. Actually, the mobility of humans is ensured by air travel who were initially infected in the outbreak areas and then traveled to other areas around the world, promoted fast dissemination of the virus. Indeed, in 2002, during the first SARS epidemic, about 1.5 billion scheduled passengers traveled worldwide. During the onset of the next MERS epidemic in 2012, it rose to 3 billion, and last year we had 4.5 billion. And this year, without the COVID pandemic, we were expected to have 5 billion air travels around. Well, to this day, 180 countries and territories were affected with novel coronavirus, a total of 12 million cases confirmed, and unfortunately 550,000 global deaths. Well, about 1/4 of all cases occurred in a single country. One potential transmission route that initially escaped the attention of the health authorities worldwide is the fecal-oral route, and specifically through wastewater, in spite of detection of this in the stool of infected patients. Well, the current seminar is indeed dedicated to this thematic. Well, I think that now I'm going to give the floor to myself again, and we'll talk about SARS-CoV-2 in the aquatic environment, what we know so far, and this is some sort of introduction to what my colleagues, my co-speakers today will talk about. So the aquatic environment covers 75% of the surface of the planet, and more people live close to major inland waterways and coastal waters. Many of the potential animals known as carriers of corona zoonotic virus, this one and the previous one, live close to those areas and eventually in close contact with humans. And this is particularly critical since we humans have been prone to destroy the regional environment where the wildlife used to live. In the case of the novel coronavirus, the viral particles may reach through the respiratory tract other systems in our body, such as the digestive, and be subsequently back to the environment through defecation. Therefore, treated and particularly untreated wastewater may be potential a conveyor for the dissemination of the virus in the aquatic environment. And we are going to discuss this pathway during this session. All coronavirus are enveloped and share similar structure features. Eventually, they may behave similarly once in the aquatic environment, and their survival as a whole particle or just their RNA in water can potentially contribute to viral spread and cross-species transmission. The fate is yet to be understood, and the water environment as a potential reservoir duly assessed. Increased temperature, as it is known, that has a deleterious effect on the survival of coronavirus, as well as UV radiation, both natural and artificial, particularly UVC. However, a high organic content of water, both particulate and dissolved, as in the case of wastewater, may increase the survival through sheltering against the virucidal effect of light. Moreover, the high protein content typically found in wastewater is thought to have a protective effect on virus survival. Therefore, the interactions with other biota in the aquatic environment, both micro and macroorganisms, including, for instance, birds and mammals, require further study. This is a major issue in the sense that some organisms may release virucidal molecules, while others may concentrate virus, like clams, for example, and behave as a potential route for humans through the food chain. And this is clearly something that skipped attention so far. Presently, fecal shedding of SARS-CoV-2 RNA from COVID-19 patients has been well reported so far, and therefore the quantification of the virus in wastewater allows the ability to monitor the prevalence of infections among the population. Indeed, environmental surveillance data reveal that members of the community were shedding SARS-CoV-2 RNA in their stool even before the first case were reported by the health authorities. Even the International Monetary Fund recommended the use of alternative methods to detect infection clusters, such as testing for the virus in sewage systems. Therefore, the concept of wastewater-based epidemiology that we are going to talk today is a valid approach to tackle the problem, since wastewater surveillance can be used to understand the prevalence of the virus in a population and has an early indicator and can serve as an early indicator of infection with a given catchment community to the wastewater treatment plant. Moreover, the current experience shows that most people will not be tested at all due to economic and practical constraints. Therefore, looking at wastewater for the new coronavirus is indeed the right way to estimate the total number of infections in the community. Briefly, the wastewater-based epidemiology approach to test for SARS-CoV-2 has notorious advantages over testing the public. This doesn't mean that we should do only this. But, and the reason is, since wastewater represents the overall status of a population within giving wastewater treatment plant watershed, as I just said, allowing the identification of temporal change. And this is particularly important when dealing with the still to come second wave. Nevertheless, that of course we hope that this will never happen, but nevertheless, the quantitative relationship between RNA densities in sewage and human infection prevalence is dependent on a myriad of spatial and temporal variables. It is becoming, however, it is becoming increasingly evident in the scientific community that wastewater surveillance can be a powerful tool to monitor the circulation of the virus in a given population, with special relevance at the community level, improving the response capability to tackle the problem and the asymmetries by the competent authorities. Most of the published research carried out since the finding of the presence of the virus in wastewater shed light on the infection prevalence within a given community as a comprehensive way to estimate the total number of infections, given that, as I just said, most of people will not be tested. Unfortunately, little attention has been given to the potential role of treated or untreated or poor treated wastewater as a source of infection to the aquatic environment. In what bathing waters are concerned, for instance, and this is a notorious public issue, particularly in the current summer season in the Northern Hemisphere, the environmental spread of the virus and the risk associated with contracting disease in bathing waters are just being highlighted. So far, the risk of transmission to humans is clearly unclear, but the Atlanta-based CDC declared that, and I quote, "The expected risk associated with contracting of SARS-CoV-2 virus via recreational waters remains low, but it's possible." Published reports accounted that viral RNA can be persistently shed in feces for several weeks after a patient has tested negative for respiratory viral RNA. Therefore, this approach is really important. And in early March, the WHO stated in a technical brief, and I quote, "There is no evidence about survival of COVID-19 virus in drinking water or sewage." They were wrong. Nevertheless, the waterborne transmission of the virus cannot be ruled out and emerge as a particularly important subject, given the fact that sewage, especially in developing countries, may be released directly into receiving waters, as we know, including the ocean, without any treatment at all. Therefore, the question of viral transmission through contaminated water to the biota and eventually to humans through direct or indirect contact is on the agenda. Fecal oral transmission may be an important, albeit poorly understood pathway for increased exposure to the virus, knowing that about 1.8 8 billion people worldwide use fecal contaminated sources as drinking water, one cannot rule out the possibility that unfitted water for human consumption to be free from the whole virus or its RNA. Therefore, one major challenge worldwide is the understanding if the virus retains its infectious properties when released into the aquatic environment. If so, what is the risk of transmission, knowing that the poor water situation will not change in the near future in many parts of the world? Indeed, a basic precaution to tackle the spread of the disease, hand washing with soap, is out of reach of hundreds of millions worldwide due to poverty. Basically, there are no affordable soap bars around. And on top of that, the water is fecal contaminated. And I know it from my own research and experience in East and West Africa, unfortunately. Going back to the wastewater treatment plants, transport, this is another important aspect. The transport of viable coronavirus in wastewater increased the potential for the virus to become aerosolized, particularly in open air facilities. And most of them are open air facilities. Therefore, their aerosolization may pose an important risk of infection of a potential high number of people within and around the vicinity of the premise. Approach the end of this talk, I would like to highlight one more unanswered question related to the fate of such new waste plaguing our environment, particularly in urban areas, a little bit around the world, everywhere. Surgical masks and other plastic materials such as gloves. It is known that the virus is able to survive for several days in such materials. After all, they may end floating in the aquatic environment and in freshwater and brackish and in seawater and eventually becoming part of that terrible thing that it's marine debris floating around the world. Are humans at risk to direct contact with such contaminants? That's one of the questions that we can pose. And also, are aquatic mammals at risk now from human to animal infection? Indeed, in our coastal urban areas, it's not uncommon to see, and I have them here around because I'm on a coastal city, to see the seagulls playing with ill-disposed masks and gloves. They love it, it seems. Another central aspect is related to the harmonization of sampling, quality control, and analysis methods for water, wastewater, and biota studies. Several analytical techniques have been used and are in use worldwide, either for concentration of samples or for the molecular screening of the COVID RNA. And particular attention should be given to this aspect. Therefore, I really think that laboratory intercalibration exercises with appropriate quantitative controls and standards are in dire need to ensure data comparability. As a conclusion, after 7 months of excited research worldwide, we badly need to gain a better understanding of the risk associated with contracting the virus through the different compartments of the water environment, and the health authorities should work shoulder to shoulder with us, with the scientific community in the development and implementation of policies in order to protect our health. Thank you very much for your attention. Now I give the floor to the next speaker, Dr. Erica Geddes.
Great. Thank you very much, Dr. Bordalo. Well, thanks everyone for joining us today. We're here to talk a little bit about how the state of Utah in the United States is using the monitoring of wastewater as part of the state's response to the COVID-19 pandemic. I'm going to talk about our broad statewide effort to deploy this tool, and then Dr. Wiedis will talk about the methods that we're using, and Dr. Vanderslice will talk more about the public health response and how this work can be integrated into that response. So the concept is relatively straightforward. I'm sure most people on this call understand what we're doing. Basically, collecting a wastewater sample at the influent to a wastewater treatment plant allows us to collect a integrated sample of a community and get a sense of the prevalence of the virus and any changes and trends in that prevalence over time. So we started our efforts here in Utah with a pilot study. And for those that haven't traveled to the Western United States, you can see here on the map where Utah sits, the state of about 220,000 square kilometers, about the size of the UK. We have about 3.5 million people in our state. And you can see here on the map the various wastewater treatment plants that we selected for our pilot study. We selected 10 facilities that represent about 40% of the population. Utah is both an urban and a rural state. We have a large urban corridor here where you can see a lot of the wastewater treatment plants are congregated. And then we also have a lot of rural areas of the state that are captured in some of these other areas. So we decided to test this idea out for a 6-week trial period, collecting samples twice a week at each of these facilities to see if we could correlate the findings with known infection rates in these sewer sheds or in these communities, and then also to see if this information would prove useful to our public health partners in the state that are responding to the pandemic. So I'm going to go through our findings from the pilot study and then talk a little bit about our next steps to deploy this more broadly across the state, and then some tips for those that want to try and do the same in other regions of the country and world. I'm going to leave the laboratory methods to Dr. Ritas to talk about. We are using the RT-qPCR methods. One thing I will note, we have 3 universities involved in our project. University of Utah, where Dr. Vanderslice and Dr. Ritas worked, Utah State University up in northern Utah, and Brigham Young University more in the central part of the state. And that's really allowing us to capture samples from a larger number of facilities. University labs, of course, can't run hundreds and hundreds of samples, and so we're somewhat constrained by laboratory capacity, and it's been great to have 3 universities on board. We did take the opportunity during the pilot study to standardize the methods across these 3 universities, and I think that's been really critical to making sure that our results are comparable and consistent, and therefore useful to public health. I'll show some of our results, but a few things about data management. We felt that it was important to correct our data for flow and population. So many of the studies I've seen in the literature report out in units of, virus copies per milliliter of water. Because we have so much variability in our wastewater treatment plants in terms of dilution and due to infiltration and just the amount of water that various parts of the state use, we corrected for that flow so that we are reporting out in units of per capita virus copies. So some of the key findings from our pilot study. First, we found the virus in all 10 of the facilities that we sampled, and in 64% of all of those samples. We saw a large increase in 2 areas of the state in late May, just ahead of an outbreak. So we were able to show that this tool can be useful to actually perhaps identify an outbreak ahead of clinical tests showing the same. And I'll show you a graph of that just here in a second. We also found, as expected, the highest concentrations in urban parts of the state, but we did find that the concentrations were higher in the 2 tourist communities that we sampled compared to other communities of similar size. And in Utah, we had our first outbreak in our ski area, which is a popular destination for tourists from around the world. The virus was not detected in the effluent leaving the sewage treatment plants, which is important and consistent with other studies. So just a couple thoughts about the potential use for this information, and Dr. Vanderslice will talk about this in a bit more detail. So you can see here on this graph the case counts in the orange line, and then our wastewater data in these red and purple dots. And you can see for these 2 communities, Logan and Hiram, which are both located in Cache County in Utah, you can see that we saw a 10 and up to 50 times jump in virus concentration in late May. And you can see that that just preceded the increase in the case counts. So we saw this obviously after our pilot was completed, but this has been a good opportunity to demonstrate to our public health partners how this tool could be used to identify kind of an early detection of rising infections. Another way in which we're considering the use of this tool is in targeting community level monitoring. So in this case, the sewer shed that you see here is captured by one wastewater treatment plant, but because of the way the sewer lines come into the facility, we're able to get samples for multiple communities at one location. And you can see here that we can actually see the differences in virus concentration in some parts of this Salt Lake County, the most urban part of the state. And these are consistent with the infection rates that have been reported in some of these areas. Finally, we think there's a useful application here in confirmation of low infection rates, especially in areas that are maybe more rural, where clinical testing is difficult, either because of resource constraints or because the population is less willing to be tested. Here, two rural parts of the state, you can see were very low or non-detect concentrations of virus throughout most of our study, with a few exceptions. And we think this could be helpful in providing the public assurance and public health officials in areas where we think there's very low infection rates to actually confirm that that's the case. So following our pilot study, we worked with our unified command here in Utah and the Department of Health and our 13 local health departments to think about how to expand this work as an ongoing part of the COVID-19 response in our state. And so we've expanded. We just started this on Monday of this week. We're sampling weekly at 30 treatment plants that are greater than 1 million gallons a day. That's about 10,000 people. We've also identified 10 more rural treatment plants that are smaller than the 1 million gallons based on a number of factors, including locations that have, or communities that have meatpacking industries where we know that's a potential for outbreaks. Some of our tourism communities, we have 5 national parks in southern Utah that attract people from around the world. And we also have some communities that have records of lower health outcomes, and that's measured as a health improvement index. And so we're trying to capture those less privileged communities to make sure that we're able to see if there's an outbreak early. Collectively, these capture about 80% of our population in Utah. As I said, we're collecting the samples weekly at these 40 sites, but we have capacity within our 3 labs to do an additional 20 samples per week, and those are being guided by our public health partners. So for example, in the northern part of the state where I showed that outbreak earlier up here, where we see an outbreak, we can spend some of those samples by getting more frequent sampling and or more detailed sampling at the community level. Our budget for a 3-month effort is $275,000. That translates to about $220 for our urban samples and almost double that for our rural samples. I'll say that one of the most expensive parts of this effort is the sample collection and transport. Just getting these samples from all of these facilities to the 3 university labs is quite an effort. Nonetheless, as others have pointed out, this is still a very cost-effective way to get broad community-level surveillance. We estimate it's less than a penny per person per sample in our urban areas and about 10 cents per person in the rural parts of the state. And that's in comparison to a clinical sample that can run in the hundreds of dollars per person. So I thought I'd end with a few planning considerations for those that are thinking about deploying a similar level of use of this tool. So some things to think about, because we've really learned a lot of lessons in our efforts here. The planning and logistics of an effort like this to capture broadly 80% of a population of a region or state is a lot. So we have these 40 facilities more than 600 kilometers apart. We've had to hire temporary staff to effectively drive the samples around the state. We've had a lot of discussions about composite versus grab sampling. Composite sampling is preferred and not a problem in most of these red dots, which are our larger facilities. But in some of the rural lagoon areas of the state, it's very difficult to get a composite sample that would have to be collected manually. There's holding time considerations, and Dr. Wiedas can talk a bit more about those, but we want to get these samples as quickly as possible on ice and to the labs for filtration. And laboratory capacity was a key constraint to sample design. We've had a lot of inquiries since we started about whether we could get down to a neighborhood scale or even sample some of the long-term care facilities in the state or the universities in the state. And the more that you expand this down to a smaller scale, it's very, very quickly the number of samples expands. And so if that continues to be of interest, we may have to look at additional laboratory capacity in commercial labs. Part of our planning consideration was also how we would explain this data and use the data, make it relevant for public health. So we are looking at identifying triggers for where public health might take an action. This includes creating statistics where you can see a clear increase, decrease, or plateau of samples over time. Most of those statistics in the COVID response are based on daily clinical data, and we here are looking at weekly data. So there's been some work thinking about how to create those statistics in a meaningful way. We've also been talking about how to report out percent change versus absolute change. We're not at the place, at least here in Utah, where we feel comfortable connecting our data with actual infection numbers of people with COVID-19, but we are comfortable seeing the trends and helping that be one of the tools that informs public health. And there may be different ways to interpret the data in urban, rural, and tourist areas. For example, because we're converting to a per capita unit, in some of our areas like Moab, Utah, some of these areas that get a lot of tourism, they can see a doubling of their population during some parts of the year. And so making sure that we're properly correcting for that increase in population is important. Finally, I'll show you our dashboard here in just a quick moment, but we have, as I said, converted our data to per capita units to account for the infiltration and inflow. We have about a 5 to 10 time difference across the wastewater treatment plants in terms of per capita water use. And that's largely because of groundwater infiltration into our sewer systems, especially during spring runoff. So we wanted to correct for that. We found that mapping the sewer sheds was a really critical component to connecting the dots between the wastewater data and the recorded case counts. Our Department of Health was able to take the mapped sewer sheds and calculate daily case counts for each of the areas of the state that we're sampling. We've been using an online data spreadsheet for the 3 labs to report their data. We think that increases the efficiency and minimizes errors in reporting this information quickly out to public health and to the public. And then the data dashboard provides a seamless display of new information to public health officials. So I'm just gonna really quickly show you that dashboard, which is still in progress. So one of the questions here is, how do you make the connection? We've got very busy public health officials, across the country, and especially in here in Utah as well. So the data you can see here, we are showing at a statewide scale. You can see the red dots indicate where virus is increasing, the green where it's decreasing, and the yellow where it's been relatively flat. If you zoom in on any of these, you can see the sewer shed or the areas that are served by that particular facility, and then the data for that facility is plotted over here. And then we have it again, we have it also plotted against our infection rate, which comes from the Department of Health for that sewer shed. So that really helps us in real time make the correlation between these two things. Okay, last slide, if I might. So a couple tips for those that are wanting to deploy or operationalize wastewater surveillance in a region. We found that a pilot study is a really effective demonstration of the utility of the tool. and can help work out the logistical and methodological issues at a relatively low cost. It's critical to involve public health partners at the beginning of these efforts in the design of the plan. There's a lot that they can offer and ask. However, we've been very careful to minimize the demand on their time. They are all working, 12 to 14-hour days right now. And this is an opportunity for scientists from other areas of the state to contribute to the response without demanding too much time from public health. I would say one of the keys here in Utah was the partnership with my agency, the Wastewater Regulatory Agency, to help with the coordination. We have relationships with wastewater treatment plants around the state, with universities, and with our public health partners that we work with routinely for a variety of reasons. So we were able to sort of bring together those three entities. Laboratory methods should, in my opinion, focus on delivering consistent and comparable data to track trends and broad patterns. That's been the focus of our effort here, not necessarily to get the most accurate or precise number to connect to infection rates. We're looking for broad trends. Communication's key. We've been coordinating frequently, weekly, and sometimes more with all of the wastewater facilities, the scientists at the universities, our sampling staff, and health officials. And the data dashboard, we think, is a really valuable component of that communication. And then finally, as scientists, we like to sort it all out ahead of time and then go forward. In this case, because we're learning as we're responding, it's important to be prepared to adapt to changing requests from public health and the new science in this field. So not letting perfection be the enemy of the really pretty good. That's all I have for the group. And I think we're probably deferring questions till the end. So I'll stop sharing my screen and I think move on to the next presenter, Dr. Wida.
Yeah, so President, I give the floor to Dr. Jennifer.
Okay, so Erica set me up well. My goal now is to talk about the methods that we use to help support the testing here in Utah. And then Jim or James Vanderslice at the end will talk about all of the data we've generated and how that correlates to case counts. So with respect to methods, you saw a figure similar to this from Erica. There's a couple of extra things I want to point out here. This is the concept of wastewater epidemiology and looking at sampling an entire sewer shed versus individual lines feeding into that overall main sewer line or main sewer shed. So here we see most of the data that we're reporting to the state comes from sampling at the location number 1 here that feeds into the wastewater treatment plant. That is a consolidation of all of the wastewater generated from all of the people, including businesses, manufacturing industries, and individual households that feed into that treatment plant. But we also did some sampling within the sub-sewer shed at locations, say 2, 3, and 4, which could represent areas where there are a few infections, or in the case of City C here in line 2, where there's no infections. And then finally, we did do some sampling at the effluent from the treatment plants, or location number 5. So my goal today really is to talk about how we did sample collection, a little bit on how we did processing and analysis of the data, show you some results on our analysis of the virus in solids versus the wastewater samples, or I'm sorry, the water or supernatant samples. We'll talk a little bit about our inter and intra laboratory replication between the 3 labs. And we'll talk about some results of the sewer shed and sub-sewer shed sampling, and then show you some data on what we found with respect to decay of the SARS-CoV-2 RNA during storage from temperatures from 4 degrees Celsius all the way up to 35 degrees Celsius and at minus 80. So a busy slide here, but let me walk you through it. This basically shows you our entire process. Starting from the left-hand side in the blue, we see our sample collection. We primarily did 24-hour flow-weighted composite samples from the larger facilities that were refrigerated samplers. Occasionally at smaller facilities, we did do grab samples. We actually did in those facilities grab samples from a grit chamber, or the first place in a treatment plant where you started to get well mixed samples, rather than a grab sample from the influent main pipe coming in. We wanted a little bit of homogenization of that sample if we could. When that sample was then double containerized, here we do view that as a biohazardous waste. So double containerized on wet ice, and most of those samples were received in the laboratory between 2 and 8 hours after collection. Some of them were as short as half an hour, but on average it was about 2 hours after it was collected. When it gets to the lab, which is shown in the orange box here, we had the option of spiking it with a surrogate virus, the marine or mouse hepatitis virus. My lab is still optimizing that and coming up with a multiplex PCR so we can detect both that virus and SARS-CoV-2 at the same time. But as we move forward over the coming weeks, we will roll it out across all 3 labs. We then had to inactivate the SARS-CoV-2 because of biosafety protocols at our university. While the literature currently suggests the virus is probably not viable in the feces, there is still some debate about that. So out of an abundance of caution, our first step when we get to the lab is to take that bottle and inactivate the virus by heating it at 65 degrees Celsius for 1 hour, which has implications for potential loss of the viral signal over time, which is why Erica said, and I'm going to reiterate, whatever method you ultimately end up selecting for analysis in the wastewater, be consistent and use that method for all your future samples. We scientists like to tinker in the lab and make it better. Here I'm going to argue, because we're using this data to help public health folks, once you have a method that seems reproducible, stick with it. And later on determine how much of the virus might have been lost and what your minimum detection limits are because of those losses. Okay, so after inactivation, we then will centrifuge the sample to try to remove some of the solids. And we'll do a pretty gentle centrifugation, about 4,000 G for 20 minutes. We'll then separate the solids and liquid. The liquid supernatant over the top will be acidified down to about a pH of 3 to 3.5 with hydrochloric acid. And that'll then be filtered 3 times. So one filter we archive, stored at minus 80 degrees Celsius. And the other 2 filters, which get about 100 mils each, those are 0.45 microns mixed cellulose acetate fibers. We then will take those directly into an RNA extraction. That RNA extraction can be with a commercial kit or can be with a manual method, like a Griffiths 2000 paper in AEM that works pretty well. We'll then do a triplicate qPCR. We'll also have a positive and negative control, of course. If you have questions about this particular method, and I think I saw some questions come up in the chat about, well, what's the best method? What is going to give me the absolute numbers? Again, I'm going to argue you don't need absolute numbers. You just need a reproducible one that you use for all future time, and then later on determine how much was lost at each stage. But Warish Ahmed recently published a paper in the Science of Total Environment, Stoughton, in 2020. You see the DOI number there below. I encourage you to take a look at that. He did a really good job of evaluating a couple different methods. The one you see here is, I think, method A, and he estimated when he used the MHV surrogate virus for the SARS-CoV-2, there was about a 26 plus or minus 15% recovery of that. The best method he ever tested only had a 65% recovery of this surrogate MHV virus. So there's no one perfect method. Again, pick one, make sure it's consistently used. Okay, then finally in the green box is the analysis. What we get out of qPCR is, gene copies per milliliter or liter of wastewater that you collected. We then convert that into viral gene copies per capita within the sewer shed, so people within the sewer shed, and then per day. And then we're going to report that out to the health departments, and they're going to put it up on a publicly available dashboard. Okay, so some results. This shows you for the several samples, so 5 different samples, actually it's more than 5, this is the number of facilities we did this test at. We tested for how much of the virus was detectable in the solids after centrifugation versus the supernatant that was on top of that centrifuge solid sample and that was retained on the filters. And by and large, there's a more RNA that we detected in the supernatant samples than on the solid samples. Keep in mind, we were only centrifuging about 200 mils. Influent tends to be not as many solids, for instance, as a return activated sludge sample. Other people have recommended doing return activated sludge samples. We're publishing papers on that. That might work. I would worry about inter-utility or inter-facility comparability between those results, because, you know, how often they waste the sludge might differ from different facilities. The pre-treatment before that might differ between facilities. So that's why we focus primarily on the influent to all of the treatment plants across Utah, rather than trying to focus on solids somewhere else within a facility. Here we see some results of our inter and intra laboratory replication. We did this for 3 different facilities. You'll see on the left-hand side of all 3 of these plots are the viral gene copies per milliliter of wastewater. Across the bottom, you see the different labs. So we had 3 labs, lab 1, 2, 3, and we all got the same sample from each of the different facilities. And we did 3 filters from that one sample we received. And we then did triplicate qPCR from each one of those. So this, for instance, facility B, we did an analysis, an ANOVA analysis of the means, and we did not find any significant difference between the different laboratories. Same story for facility C. We did see some variability in facility A. So what we did with these results was basically moving forward, we are going to be doing duplicate filters from every sample we get in all 3 of the labs, and triplicate qPCR, including matrix spikes, to make sure that we're not having any inhibition in the analysis. And then those averaged values will be reported to the public health partners. So replication is key, and make sure that if you have multiple laboratories across your area doing testing, that you make sure that everybody's got similar results. Okay, so now let's talk about some results from subsewer shed or interceptor sampling. So you see 2 plots here, both from the same location. On the right-hand side, in the box is the location for the main treatment plant in the star. CVWRF is the name of the facility. Each of the colored areas are sub-sewer sheds that feed into that main facility, labeled A through G or H. And then the main line that feeds into that facility is shown on the figure on the left-hand side in the red lines. So those are the main sewer lines, and the points that the facility samples could collect samples that feeds into the main CVRF influent. Okay, the key point here is, so the utility, Central Valley or CVWRF, samples on a monthly basis the amount of water coming from each of the individual entities, because that's how they charge the cities for their rates, okay? So we know the amount of water coming in from each one of these different facilities, and they also have 24-hour composite samplers there, which was great for our analysis. Okay. But because there are such different flow rates from each one of these cities, and I'll show you in a table what those different flow rates were, and different populations in each one of these cities, what we ended up doing, as Erica suggested before, was converting all of our data from the gene copies of the virus per liter of influent that came into the treatment plant into this viral gene copies per capita per day. And we did that taking into account the flow rate into the facility or from each sub-sewer shed, and dividing that by the population in the sewer shed. So this took a lot of GIS work for folks to marry up the sewer shed maps with the population maps, and ultimately with the public health case counts within each one of those areas. Okay, so a very busy table here. Let me walk you through it. In column A, we see the different facility, either the influent to the main treatment plant on the first row, or each one of the individual entities that fed into that main treatment plant. The flow rates are shown here in column B. So Central Valley treats about 51 million gallons per day. Apologies to the international folks. That's the units we use here in the United States. So average and standard deviation of flows are shown here in column B. The population served in each one of those, this main sewer shed or the sub-sewer sheds are shown here. So about 5.5 500,000 people are served by Central Valley. And then the gallons per capita per day are shown here in column D. On average, in the United States, we have about 100 gallons per person per day, and that's shown here for Central Valley. But you'll see that some of the smaller cities that feed into this main plant have lower flow rates. For instance, Kearns, the KRN interceptor only has about 61 gallons per capita per day. And other ones, for instance, the second line from the bottom, South Salt Lake City, has a terrible problem with their sewer lines, and there's a lot of infiltration such that you see 334 gallons per person per day coming into that plant. Okay, so column E would be how many samples we tested. So we tested 25 influent samples to Central Valley, and 96% of those were positive. And we did 4 tests from each of the other sub-sewer sheds that fed into Central Valley. And then the 3 columns on the right-hand side show you our qPCR results and the converted results, and then the case counts. So column F shows you the viral gene copies per liter, average and standard deviation for all of the samples that were tested. We converted that into millions of viral gene copies per capita per day in column G, and then the column H shows you the COVID-19 confirmed case counts per 100K. So again, I emphasize there is different flow rates for some of these facilities and sub-sewer sheds, so make sure that you account for that I&I in your calculations. Again, we saw a high % positive in the Central Valley samples, not surprising. This serves the main part of Salt Lake City. And the certain sub-sewer sheds accounted for a significant amount of the SARS-CoV-2 signal, and that correlates in some cases with the case count. So for instance, for the Granger Hunter area here that I highlighted, high gene copies per liter in the wastewater and also high case counts. Other ones, for instance, Kearns with a low flow rate also had high case counts, and so did South Salt Lake City. Okay. Here we see the same data, but I put it on a bar chart because it might be a little easier to compare these numbers with the influent to the treatment plant. So the bar charts on the left-hand side show you the data in 2 different ways. We sampled the interceptors or sub-sewer sheds on 4 different days, April and May. The left-hand plot shows you millions of gene copies per capita per day. So those bar charts for each of the individual sub-sewer sheds show you the amount of virus, which accounts for the population and also the flow rates. And then the 2 bar charts in plot B on the right-hand side show you just the gene copies per milliliter of wastewater that you might have gotten from qPCR without accounting for flow rate or for per capita. So it's important, and we're going to make this point a couple times, I think, that you do account for per capita and flow rates. For instance, for the South Salt Lake City, the purple ones set on the bar chart, plot A, on the left-hand side is significantly greater proportion of the SARS-CoV-2 signal compared to when the purple for April 13th, if you just look at gene copies per milliliter of wastewater. So for instance here, here versus here. The other thing that's important to note here is, you know what, we don't see all of those bar charts virus signal adding up to what's coming into the main plant. So the open circles or the white circles represent the concentration of the virus coming into the main treatment plant. In an ideal world, that circle would be at the top of those bar charts. So why is this? Why do we think this happened? And in part, we think it's because of the I&I, but also because of decay of the virus as it travels through the system. So we see that here in part. We took some of the samples from 2 different facilities, and when we received them in the laboratory, prior to inactivating them at the 65 degrees C for an hour, we just took the bottles straight from the facility and we stored them between 0 and 24 hours at a variety of temperatures. So we stored them at 4 degrees Celsius. We stored them at 10 degrees Celsius, which is similar to the sewer temperatures that we see here in Salt Lake. and we stored them at 35 degrees Celsius, which would be much warmer, but might be seen in other parts of the world, or maybe even in a lagoon system where you have a warm, sunny day. The take-home message is we're seeing a lot of decay of that viral RNA over time. At 35 degrees Celsius, we saw a minus 0.18 per hour first-order decay rate of the viral signal. At 4 degrees Celsius, you're going to get about a 2 log reduction in the virus signal over 24 hours. A similar story at the 10 degrees C. So this leads us to our overall recommendations. Get the samples to your labs as soon as possible and have the shortest hold time possible prior to that RNA extraction so you don't lose signal. We recommend that you account for flow rates to the facility to understand the viral loads within the population. And that sub-sewer shed sampling can be very valuable to identify regions with higher SARS-CoV-2 signal, but you do need to account for inflow and infiltration and potentially industrial inputs which may decrease the signal over time. Okay, with that, there were a lot of people that helped with this project. Great coordination by Erica Gaddis's group with the Department of Environmental Quality, and certainly all the facilities. And then our colleagues at Utah State University, Dr. Keith Roper, and at Brigham Young University, Zach Andrew. And with that, I will turn it over to him.
Yeah, so the floor now is to Professor Dr. James Vanderslice. Sorry, I misspelled your name, Vanderslice, sorry.
That's all right. Good morning. Just wanted to extend our appreciation to MDPI for setting up this, this webinar. We look forward that it will start a dialogue because this is an area that's brand new. We're all learning about it. I also wanted to really point out the efforts that particularly Dr. Wheatus in starting this right off the bat. As soon as this problem started to become evident, she really jumped on it, using her own funds, getting sampling started, talking with our Division of Environmental Quality and Dr. Gaddis, who then grabbed it and started rolling. So it became a great team really fast. And like, you know, a lot of things with public health, dealing with anything that's infectious, speed is of the essence. So I wanted to go through and reiterate somewhat what are some of the potential public health applications, then take the initial results that we have, and from that start to look at what does this tell us in terms of being able to use wastewater monitoring to meet those applications. And then given where we are now, you know, what are the implications for, you know, how can we use it going into the future? Now, some of these ideas are ones that you've seen brought up by other authors. Some of these were presented by Dr. Gaddis. Let's go through some of the main applications. First, there was a lot of interest and I think hope that you could actually be able to predict a prevalence or cumulative incidence rate by looking at concentration of viral particles, virus DNA in a sewer shed coming into the effluent of a plant. So that's looking at an actual mathematical relationship where you could predict the number of infected individuals. Second is basically being able to identify which areas have higher or lower disease burdens. With this, it's a little bit perhaps easier in that you're not needing to have developed a relationship for each sewer shed, but being able to use information within each sewer shed and make some comparison between them. The third is really being able to present evidence of low prevalence. So this is really kind of a subset of the first one, where at least you may not be able to say, how does the prevalence increase with the increasing gene copies per milliliter or per capita, but does 0 equal 0? when you're not seeing any viral particles, not seeing any viral DNA, RNA, are we actually at a zero? And then looking at things over time, do we see trends? Can we find an initial outbreak where you're starting with a place where it has a low background? Or on the other hand, can you establish that we are seeing reductions in prevalence of both not only symptomatic cases, but the asymptomatic and pre-symptomatic cases which still are shedding viruses into the sewer system? So looking at the first, these are all of our results. And this is looking at daily case counts associated with each day that we did sampling. And we have looked at some information looking at lags, and pretty much the relationships are the same. Each of the different geometric symbols represent a different wastewater treatment plant. You can see that we have this as gene copies, millions of gene copies per capita per day against case counts. One thing to notice is just we have a lot of observations where we have really low numbers of daily case counts. Our populations throughout these sewer sheds range from about 10,000 up to the highest being the sewer sheds and sub-sewer sheds that Dr. Wheatus was presenting, where that is about 500,000 individuals. And that's the open circles. But there are a lot of observations that are essentially zero. At the same time, we were looking at, in terms of millions of gene copies per capita per day, a range over 4 orders of magnitude, from just above 0.1 all the way over 1,000. Which gives some pause in being able to say that if we have, no cases, are we having a lack of a signal? The other thing you can see is there's a lot of variation between the different systems. Now, the ones where we're having high case counts, they basically derive, somewhat of a linear relationship with the log of the gene copies per capita. And the open circles and the black squares are really 2 of the largest sewer sheds. The black squares is Salt Lake City, the capital city, with about 210,000 population. So the idea that there's some, common or universal relationship here certainly doesn't seem to be borne out, and I don't think was really expected given all the differences between the different sewer sheds. Now, one thing that's a benefit of having a webinar is you can put a lot more on a slide and expect people to be able to see it. This is for each of the wastewater treatment plant influent measurements. And what it's looking at is on the axis on the left side is the millions of gene count per capita per day. On the right side are the number of new COVID cases in that week per 100,000. And then it's going across the different weeks of the study. The vertical dotted line indicates the first week where we started sampling for that facility. And the red line, of course, is the new cases per 100K population. What do we see here? These first 2 are Central Valley. That's the system that serves about 500,000 people. It's the system that Dr. Wheatus was presenting and showing the sub-watersheds for. And then this is Salt Lake City. These are the 2 that really have had high numbers of cases at times and were really driving any bit of an overall relationship between what we're seeing in terms of gene copies and what we're seeing in terms of health outcomes. A lot of variability, no clear patterns. These are all weekly averages, so they tend to be for essentially 2 samples within that week. If we move down, again, you can see a lot of different patterns. Patterns where we do see a number of increases as time goes on, and at the same time, we do have some indication of increases. These two areas, Logan and Hiram, they're both unique in that they were up north and very low case counts up until about week 8, week 10, which is the end of May. And this is where there was an outbreak. A lot of it was associated with a meat packing plant, but there is more of a relationship. Although here we're seeing a signal and perhaps a small signal in terms of the number of cases. In these systems, a lot of these systems are a lot smaller. You can tell just by looking at the scale of the cumulative incidence of cases. But in this case, where it's our case at an area that encompasses Park City, which is a big resort area, gets a lot of outside people that come from other parts of the United States in for vacation. And this was really the area in Utah that had the initial number of cases. This is kind of where you can say a lot of it began. And that they instituted really strong measures really early and were able to drop their case counts, and yet we can see that there's a decrease. This again is looking at the millions of gene copies per capita per day against the weekly cumulative incidence. And here we have it lagged. And this is what, at least in the US, we would refer to as cherry picking, in the sense that here are the results for where we saw a relationship. Here we have it not in a log scale, but in an arithmetic scale. And you can tell there are some differences, but generally they're across the same scales. Now, yeah, we have a row of 0.82 on a Spearman's correlation coefficient, but what's that being driven by is a bunch at 0, and then we have an increase. And in each case, that comes through. For this case, we do have a couple of other high levels. So if we look at these in more closely, this is looking at Logan City where there was the outbreak. Here's the graph I just showed you. Here's the one that I showed you before, looking at it over time. And clearly, this data point is this data point where we had that outbreak beginning. So when there is a low level and it increases, A, we're able to catch it, but B, this is what's driving for our data, which again, time-wise, it's over a relatively short time frame, driving any really observable relationship. Hiram City is just south of Logan. Same influence from the same outbreak. Again, 2 points that really aren't essentially 0, and you can see those. This is one, that's the other. And so being able to see that, when we're comparing things to 0 or to very low levels, you can start to see a relationship between gene counts and cumulative incidence. And the last one we were able to see a significant correlation was with East Canyon. Again, this is Park City area, the resort area where it all started. We have a few higher counts. And again, you can see this is the opposite case where we're starting high, but we're starting to see that drop. So some observation on this, clearly there's no common relationship between the gene counts per capita, number of cases, looked at this in a couple of different ways, and that there's large variability in the viral gene counts per capita when you have low case counts or low cumulative incidence in the different sewer sheds. and that you see different patterns in the different sewer sheds when you're looking at both gene counts, COVID cases over time. No strong associations between the two for most of the sewer sheds, and really some inconsistent short-term covariation. And this is what we saw with the site-specific graphs looking at gene counts and looking at COVID cases for each week of our study. We did see some temporal relationships, again, when there's a large increase in cases starting from a low baseline, where there's a large decrease in cases down to a low level. And again, it becomes easier to compare 0 or really low levels to something other than really low levels. What we're working on now, this was really our first cut, is how do we incorporate data that measures or is proxies for the other factors that we know would affect viral gene count concentrations at the bottom of the sewer shed. I saw one of the questions come up as to whether we use population or population equivalents. We use population. We did have really good population counts because we have a statewide registry. We were able to geocode 97% of all the addresses for the 3.1 individuals in the state, and then assign that to each of the sewer sheds that we were working with. So taking account of non-residential population, so we can have some sort of population equivalent, and we're working on being able to look at, okay, how many employers are there and what kind of businesses are there? At what point in time were people actually working at the business as opposed to when a lot of the shops were closed early on? Same for shoppers, restaurant patrons, and this is going to be based on the number of businesses and the time frame as to when the businesses were open. Looking at water use behaviors, neighborhood income levels. Certainly you have more water use in higher income neighborhoods. We'll be able to use satellite data to look at vegetation coverage, essentially lawns. We live in a desert, and so still there's a lot of lawns that are grass lawns that people maintain, and that accounts for a large amount of water use. And then actually being able to use perhaps data from the water utilities on water use. Non-residential flow, industrial usage, infiltration, which has been mentioned a couple of times. A lot of that is going to be difficult to measure directly, but certainly by looking at per capita volume into the wastewater plant. As Dr. Wheatus discussed, decay being perhaps a major factor, and for this we're going to be looking at transit times, any of the different systems that have modeled their wastewater collection system, looking at distance from different consumers to the sampling point. And then we're comparing this to the known cases. These are, for the most part, people that are symptomatic that then go and get tested. There are a few other regions that people would get tested without being symptomatic, but that accounts for a very small number of our tests. So is there variation in the ratio between the symptomatic and asymptomatic and pre-symptomatic cases? And whether this would probably be associated with demographic factors. There's some evidence of that, but also be able to use current and lag cumulative incidence to get a sense of that. And then use this group of data with what we're seeing in terms of gene copies at the sampling points and employ more predictive modeling approaches and see whether we can do a better job of using data on gene copies at the sampling points to help us inform what's going on in those sewer sheds, both between and over time. And again, just measuring gene copies in sewage, it's one piece of information, and we need to make it as useful as possible so public health can then combine it with the other information they have, as well as, and we all know there are considerations beyond just public health in making a lot of decisions about what kind of public health interventions will take place. And I just want to spend a couple of minutes to talk a bit about, at this point in time, what are some of the implications as to how this information can be directly used. These are the case counts for Utah. Utah, again, has a population of about 3.1 million. And things started to ramp up in the United States early March. For example, at the University of Utah, it was about March 10th that we went to all online teaching. We saw this increase. Everyone was very concerned, instituted sort of stay-at-home recommendations. And since then, there was what people felt was a drop, but then it really, there's a lot of day-to-day variability. Depends on the day of the week. Pretty much we're at a consistent level, and this was at least expressed by the governor of the state that, gee, we're doing okay. We can move from a red to an orange. We can start to open up the economy because the economic impacts were becoming quite evident. And that even this drop here, right here, great increase. This was one of our major 3-day weekends, and the drop was probably more attributable to less testing. And then since then, through June, we've seen a steady increase. This is yesterday's number, the highest ever, 713 new cases. So this is the time frame of our study. So as we look at and say, well, gee, what kind of variability did we see? When we're at this point and this plateau, which was about I believe it was cumulative incidence of about 5 cases per 100,000 population, we're up at this point at about 17. So at this point, we thought, oh, wow, look at this variability. Can we catch it? Looking ahead in time, this variability is nothing compared to what kind of variability we're seeing here. And this is really now the scale that we're looking at. And so we're looking at being able to restart this and follow it forward will be really, really important. One of the applications we discussed was being able to have sort of an early warning system to be able to identify and see where we have outbreaks starting. Almost everywhere in the United States has been impacted. There's only about 10% of the 3,000 counties in the United States where there are 10 or fewer cases reported. These are rural, small population areas. And you can see this here. This is New York Times presented these data that are collected from a variety of sources. This is looking at trends. Red is where you're having a large increase in cases over the last 2 weeks. You can see the color code. A few places where blue and you actually have falling rates. Here are ones where you have few or no cases. The no cases is virtually none. There's virtually no county with no cases. But still there's a lot of areas with small number of cases. So this is, at this point in time then, how can this information be useful? We talked about monitoring for looking at increased levels in low prevalence communities. Now, low prevalence communities, again, this is rural. Is it going to be a priority, and will it be cost effective? Because there's going to be a low number of cases. There's a low number of people. Fortunately, as we were designing the next phase of our study, really we're shooting to get those some of those areas, but these are also small communities, many of which that don't have centralized wastewater collection, and as Dr. Weedis mentioned, manual sample collection. They don't use composite samplers. More important, in thinking about those areas that are blue, we're about to go, there's a lot of talk at this point of, are we going to start, the term we use here is locking down or requiring masks. To be able to support those those activities, we need to be able to demonstrate that we are seeing reductions. So I think one of the important ways is that we're being able to look at that there's evidence of a consistent reduction in transmission. And of course, if and when we get down to lower levels, you know, that early warning for the next wave. Novel applications, being able to use this to monitor groups that are supposed to be COVID-free, or we're starting out as being COVID-free. An outbreak could spread quickly. We can do control measures to quickly institute, and the consequences are high. So small special populations where it could be valuable to confirm there is no shedding, that early warning for this group, and provide assurance that the interventions that are being done are effective. And what kind of groups are these? Well, students in residence halls or athletic facilities, prisoners, residential care facilities, hospitals for non-COVID units. And this is something that we're working on currently at the University of Utah. There's a lot of concern about bringing students back, having them in residence halls, and that we're just starting a pilot to be doing sampling, wastewater sampling of individual residence halls and of floors on the hospital to look at, can this be used in a really small, you talked about a sub sewer shed, this is a very small sewer shed where we're talking about individual buildings, individual floors on a hospital. And using that kind of approach, and maybe even in tiered sampling, where we're sampling at points that we can access the sewer system that collect sewage from several buildings. If we get a positive, if we get a hit, then go to individual building monitoring. Now, seems like a lot of effort, but again, this is a population, they're bringing students in, they're screening them. These students should be COVID-free. If anyone's positive, they're gonna go into quarantine. So you could have really effective measures, and if you have an outbreak in one of the dorms, in the athletic facility, the university sees that as having a lot of consequence. Well, thank you very much for your time and your attention, and I'll turn the floor back over to our moderator.
The first one now is from Emmanuel, and he wants to ask if detection of viral RNA in wastewater settings are said to be associated with unstable as well as real-time PCR detection as said to be insensitive. In this case, detection of viral RNA in wastewater treatment plants can, the application of magnetic biosensors can be used along with wastewater-based epidemiology.
Yeah, I think the question is wondering whether there are biosensors or other methods that could be deployed to the facilities. Yeah, that's a great question. Unfortunately, wastewater influent is highly complex. There are thousands and thousands of different types of just viruses in the wastewater and bacteria and protozoa. To my knowledge, there's not currently a magnetic biosensor that would be valid for testing for SARS-CoV-2.
Well, another question by Alex. There is a statement of the question, even though there is a correlation between COVID virus viral concentration and with the general population, I have heard that virus concentration in wastewater is most likely not viable due to the passing through the digestive system, in this case, low risk of getting the virus through wastewater. Well, there are contradictory, of course, informations around, but there are already some studies, and I mentioned one that detected in the stool of COVID-infected patients. So the problem to find out if the virus is active or is alive it's related to the facilities, lab facilities. I mean, we all do this in the lab too category 2 labs, and in order to work with the live virus, we need class 3 facilities, and most of us don't have here. So that's quite difficult. When we perform the screening for SARS-CoV-2, what we do is we screen the RNA, and we don't know if the RNA it's outside the capsule, or it's encapsulated, or it's alive. We don't do the discrimination. Okay, so next question. It's from Emmanuel. How do we relate the correlation of COVID-19 virus in wastewater treatment plants to the number of infected?
Based on our work so far, just looking at those two variables, highly variable, no really consistent relationships. I don't think there will ever be an equation that can come up just looking at those. I think some predictive models could be developed, but again, as I showed, that there's a lot that has to be taken into account to look at the other factors that affect what what the load is at the sampling point at the bottom of the sewer shed, that's beyond what we know about the number of infected individuals.
Okay, another question from Adele. What do we know about the efficiency of eliminating COVID virus using water disinfectants? Well, this is a very good question, and some studies have been performed. I don't think that there is no silver bullet for this, but I will give the floor to my colleagues today.
Common methods for treating wastewater include chlorination in the United States, UV disinfection, a couple of other methods as well, but those are the two most common. Well, if chlorine didn't work, no one would tell you to use bleach to clean surfaces. So it works, and there are studies to help support that on food surfaces. I don't know, I haven't seen a study on UV, but we do know some evidence about the decay of the virus on outdoor settings that are exposed to sunlight, and it does decay quickly. But I don't think definitive studies have been published on those two disinfection methods yet.
Yeah, and actually this is quite interesting, because chlorine is well, it's used commonly in the US. It's not used so often in Europe because of the production of byproducts, and particularly nitrosamines, and that it's produced from the contact of chlorine with nitrogen bond organic matter. And so here we do other things, particularly UV, and that works, although it's quite expensive. Okay, so now a question from Andrea. Do you make tests in order to detect compounds that exert anti-COVID-19 actions? Real-time PCR is used to do such type of analysis?
Interesting question. We are not doing that kind of research right now. What RT-qPCR tests for is just the RNA. So you would not know whether it was a viable virus or an inactivated or dead virus, or just the nucleic acids. So I don't think RT-qPCR is the appropriate approach for that. You would, again, as suggested by our moderator, probably need a BSL-3 facility where you could actually work with a live virus, which is, those facilities are rare and expensive to operate.
Yeah. And since the question, it's really interesting, we must not forget that in the water, there are lots of organisms that produce molecules that have deleterious effect on virus, and these are natural things. So, but since this is so new, we are really entering a very, very new field. I think that there is time and to look at this, because it is quite interesting. We have examples for other viruses of this, and particularly look in countries where medicine it's difficult to reach. Most of the people have difficulty to reach because there are no health facilities. They use a lot of what they call traditional medicine, and some of the plants, for example, are known to have antibacterial and antiviral properties. So this is an aspect that should be looked as well. Now, a very important question from Aaron. I understand that considering safety, how safe is after 1 hour at 65 degrees? I think that the question is for Jennifer.
Yeah, that is based off of research done on SARS-CoV, the prior infectious agent. They did a lot of testing and showed that that virus was inactivated at 65 degrees Celsius for 1 hour. That's the best science we have to date.
Well, there is another question from Kelly on the methodology. Can you freeze samples and hold at -80 prior to shipping or transport? Can you ship frozen samples on dry ice and keep them cold? If you don't have a lab within a couple of hours, how would you suggest conducting such study? Okay, first of all, we are talking about virus, and as with all viral sample, we should process them as soon as possible and try to avoid refrigeration or even freezing. Of course, if it's not possible, we must find other ways. And please, Jennifer.
Yeah, that's a great question. So we did do a test. My colleague at Keith Roper at Utah State University did test the virus decay at 80 degrees Celsius. Over 1 week, he saw almost like a 90% reduction. Now, keep in mind, this is wastewater influent. It's got a lot of nuclease enzymes in there that are going to attack the RNA. So again, process them as soon as possible. There are methods for field filtering. There are methods for adding an RNA preservative. You're going to have to try a couple of different techniques if you're going to do really remote sample processing, if you can't get it to a lab within a few hours of collection.
Well, there is another question from Ayman, and this is really interesting. If RNA of the virus is decayed at high temperature, does it mean that it's less likely to spread in temperate climates? Actually, there is a report from an early report from Mexico, where they stated that because Mexico has a tropical climate with an average temperature of 29.5 degrees, I think. Well, that's the explanation that the researchers gave for the low numbers a couple of months ago. Although right now we know that Mexico is one of the highest, unfortunately, one of the countries with the highest case numbers, unfortunately. So this is very early still to understand the climate or the ecological effects on the numbers of COVID-19 cases worldwide. But as with other viral diseases, probably climate conditions have an impact. And look, for example, in Africa, right now is in the southern part of Africa, below the equator, it's the winter. And when I mean winter, it's, for example, temperatures of 18, 15 degrees during the day, that it's really cold. And what we see in many countries, although Africa is not a very good example because of it's difficult, the facilities to detect, to process samples are quite limited. But anyway, we have been seeing an increase right now at this moment in the southern hemisphere, that it's the winter time, an increase of cases. So this is another aspect that we should pay a lot of attention, because these climate and ecological aspects are probably have a lot of influence. So now it's for Dr. Jennifer, a question from Chandra. When was this experiment on virus RNA decay with hours of hold time for different temperatures? As I heard, the virus can even resist still higher temperatures. Can you reply? Thank you.
Interesting question. Again, we'll point back to our data. We did see a decay of the virus at 4, 10, and 35 degrees Celsius. We did not detect the virus at 35 degrees Celsius after basically 5 hours. I have not seen any results showing that the virus survives for a long period of time at higher temperatures.
Well, another question from this time from Anna. What gene or genes have you found to be the most reliable and potential abundant stable target for real-time PCR?
So I don't know which one is most reliable. I can tell you which one we used. We used the US CDC recommended N1 and N2 primers, which is used for diagnostic testing for patients in the hospital.
Yeah, we use the same. Well, now, question from Ashish Kumar. Supernatant acidification is process to remove COVID molecule. What technique should be used to kill RNA strains of COVID molecule in human body to save human life? Well, what I think is to drink disinfectant. It's not the right way for sure.
No, don't do that.
Okay.
We did supernatant acidification primarily to change the charge on the virus so that it would stick to the filters.
Yeah. Well, from Alamgir, what is the relationship of virus severity with winter season? Okay, because, sorry, it seems that, okay, sorry. Okay, because more deaths occur in the North Hemisphere, USA and European countries as compared to the South Asian countries while having more population. Well, here the question has two parts. First is the winter. I mean, I already mentioned that in an early answer. The other thing, it's another aspect that there are researchers looking into is how the genetics, human genetics influence the response, the immunological response to the infection. So we still don't know, but this is quite interesting. One thing, it's true. In Europe, we start with the epidemic during the winter season, at the same time as the common flu, and that was the problem. That initially it was difficult to discriminate between the common one and this new one. Now we see an increase of numbers in the southern hemisphere, so that's very interesting, and I hope that we will be able to learn as soon as possible more about that.
If I could add to that, in the U.S., again, we were starting in New York City, late February, March. It was very cold there. It seems like the behaviors and control measures really, the influence of those behaviors, people being out, not wearing masks, that really is driving the driving factors in what we see in our increase in cumulative incidence, much more than an ecologic effect.
Yeah. Okay, now there is it's not a question from Anand, actually it's a statement. Poor drinking facilities in rural tribal areas is completely right. Okay, we are sitting in the developed country, and so I'm in Europe and you are in the US. I'm in the co-speakers today, so we have tap water, so we have clean water around, but unfortunately millions or 2 billion actually almost don't have that. So even if we wash the hands with soap and water, it's highly contaminated, fecally contaminated. So the probability of having the virus as well as in the other case of other virus, it's quite high. So, and so we are probably, we already have it in our hands, but water that we are using to wash them also have. So the power of the soap, we don't know. I mean, I think that nobody knows how efficient is washing the hands with such water. On the other hand, remember that besides that, there are people that need to walk several kilometers, not miles, please, kilometers, every day to go and fetch water. And washing the hands probably is the last option for them, because carrying for several kilometers a bucket of water on their heads of 30 liters, that means 30 kilos, it's huge. So the water basically it's for drinking and for cooking. And that's why if we don't solve this problem worldwide, probably we are going to have for several months or even more hotspots a little around the world. And because this is quite new, this is a novel coronavirus with a behavior different from the cousins and from other viruses, we don't know the consequences at all. So now Christopher has a question for Dr. Waidas. For cities with combined sewer systems, viral concentrations in wastewater influent will obviously be diluted during rain events. Does this limit trends? Does this limit trend analysis to separate sewer systems?
Interesting question. I think, as Dr. van der Slijs alluded to, there's not one direct correlation between what we measure in the influent and what we see for case counts. Flow rates are important. If you have a mechanism to assess how much rainwater is infiltrating into your system, you may be able to back calculate that influence, in addition to all those other factors that Jim was suggesting you look at. What kind of facilities are there? How often are people there? How long are they there? How long does it take to travel in the sewer system? I think will be a complex model, which will come out probably in months, if not years from now. It'll take a while to answer that question.
Yeah, on the other hand, from my own experience here in Porto and in this area where I live, we have both systems. We have some sewer system that is separate, others that are not, and those that are not, as soon as it rains, I mean, there is dilution, theoretical dilution, yes, but on the other hand, we have a huge input of sewage from septic tanks, overflow, and so on. So it just eliminates the dilution effect. So, and if we have infected people, so the probability of having a higher concentration of viral particles, it's very high likely to occur. So now a question from Basim. What do you suggest to do in order to monitor on-site wastewater system, especially that most of them are not using any disinfection techniques? He's quite right.
So this is something I've heard discussed at the national level, and I think also answers one of the last questions here on the list. We haven't looked at that yet here in Utah, looking at on-site systems or non-sewered communities. I've heard some discussion nationally about looking at like large on-site systems that might be representative of a community, such as a church or other community gathering area. That could be a way in some rural areas to try and capture an integrated sample. I think sampling at the home level, septic systems or on-site systems will be probably not feasible, at least right away.
Another question from Mr. Kumar. COVID molecule has a fatty layer cover which can be dead by using boiling water drinking. Then our RNA strain can be killed in human body to save human life. Well, indeed we can be killed by boiling, that's for COVID, for other viruses and probably for COVID as well. But when we, for example, drink or contaminated water, for example, with COVID, we do have some protection in our body. It's in our stomach, it's this acid protection that we have, and eventually it can be suppressed, eliminated or suppressed with contact with the high pH of our stomach. But the problem probably it's not that one. The problem is when we enter in contact with the virus through respiration or through surface, and then we put our hands in our nose, and you can't imagine how many times a minute we do that, or in our mouth, and this migrates to the upper respiratory system. And if it stays there, okay, we can have some problems, but it's not that problematic. The problem is when it migrates to the lower, to the lungs, and then and so on. And from then it enters. It may enter through the bloodstream. It can be in contact with the organs and can go back to the intestines, and then it's why some have show at least 10% of the patients have diarrhea, and so this is quite complex. I don't know if somebody wants to add something to this question. Okay, so we go to the last one from Eunice. Has there been any work done in parts of Utah that are non-sewer?
And no, as Dr. Gaddis mentioned, it's under discussion. I think it just doesn't rise to being a priority, and it's a problem as we're looking at the work that we're starting to do at the university, is as you get to a smaller and smaller population, the variability over time becomes greater and greater. So there's challenges with sampling, with gathering a sample that's representative across time, which then makes it more representative across the population. So those things are in discussion, but they're just not at the same priority as trying to get at least small communities that are sewered.
Okay, so we have no more questions, and we ended up with 78 participants at this time of the day in different parts of the world. This is wonderful. I thank you, all the participants, very much for this about 2 hours of very, very interesting talks by my co-speakers today. Thank you so much for the questions. Some of them were answered privately, straight, and others by all of us. And again, my last words are for MDPI for this one wonderful session, and it went very, very smooth, and I have already some experience with this kind of webinar. Sometimes it doesn't work at all, or we have difficulties, but everything was perfect. Thank you very much, particularly to Ana Sanchez, that was the chairwoman for the session today.
I just want to thank, obviously, you and all the speakers for your interesting talks. Thank you very much for your time and your effort in the name of MDPI and the Water Journal. So stay safe.
Okay. Bye-bye.
Thank you.
Bye.